Hi, I'm Dr. Gina Conti, Chief Scientific Officer of Lighthouse Genomics, and I'm going to walk you through our Genovar report. First, you can see the producer and the provided strain name. Here we're looking at the report for Sacred Medicinals Nader CBD. With every one of the samples that we sequence or genotype, we assign it a Genovar ID. The term Genovar is meant to signify a genomic variety. And these genomic varieties are unique genomes. So, for example, every clone line shares a genome, and their genome is unique from every other clone line. This ID carries true information about the genetic identity of the plants, unlike the strain names. In this case, the Nader CBD Genovar ID starts with LGS because it was whole genome sequenced. If we had instead SNP array genotyped this particular sample, the Genovar ID would start with LGT. If we see this genome again, a suffix will be attached to indicate that it is not the first time we've seen that genome. For this Genovar, we conducted whole genome sequencing. Whole genome sequencing is exactly what it sounds like. We capture the entire cannabis genome, and that's about 800 million base pairs. We then pare that down to all of the positions in the cannabis genome that are variable in the population. It's these areas that carry information about the differences between genovars. The first module in our report is called Genetic Novelty Analysis. Genetic novelty refers to the uniqueness of a genome compared to other genomes. This figure shows the population distribution of genetic novelty that we've seen in our samples. The height of the vertical bars indicates the number of individuals having the corresponding value of genetic novelty, which is shown on the x-axis. And it's a relatively normal distribution that we've seen. Sacred Medicinals Nader CBD, it's falling out here, shown by the vertical blue bar in the 84th percentile. We found around 10 million variants in the cannabis genome, and we're using all of those to capture the results in this report.